We've spent a fair bit of time going over different evolutionary mechanisms that can change allele frequencies in a population. We've talked about mutation, natural selection, gene flow, genetic drift. We've also talked about non-random mating, which can't change allele frequencies but can change genotype frequencies. This brings us to the topic of Hardy-Weinberg equilibrium. A population is said to be at Hardy-Weinberg equilibrium if, evolutionarily speaking, nothing interesting is happening. That is, there's no natural selection, no mutation, no genetic drift, no gene flow, and mating in the population is completely random. If this is the case, two things will be true. First, allele frequencies will stay the same. They will not change over time. And second, genotype frequencies would be just what you expect if we were pulling alleles out of a bag. The genotypes will be in the ratios p squared to 2pq to q squared. If you flip this around, the logic is that if allele frequencies do change over time, or if the genotypes are not in those ratios, then at least one of these 5 mechanisms must be at work in a population. On the face of it, this may seem pointless. There are very few populations in nature where all 5 of these forces are actually entirely absent. And why would we really want to care if nothing is going on in a population? The reason this is useful is because Hardy-Weinberg equilibrium is a null hypothesis in population genetics. By knowing what a population should look like if nothing interesting is going on, you can see clues that tell you what interesting things are going on in a population. To illustrate this point, let's go back to an example you've already seen. The outbreak of Kuru among the Foray people of Papua New Guinea. Now, just to remind you, prion diseases rather, are diseases that are apparently carried by an actual protein, not by a virus, not by a bacterium, not by any kind of other infectious agent that carries any kind of DNA. The best known one is mad cow disease, but there are prion diseases in a number of different species, including humans. The human prion disease that we know about is called Kuru. In Papua New Guinea, it was called the laughing sickness. And when it was at an epidemic state in the first half of the 1900s, it killed thousands of the Foray people. When it was at its most deadly, 2% of the population was dying from laughing disease every year. So clearly this was a pretty powerful evolutionary event. Here are the data that I showed you in class. There's a particular gene, PRNP, and at codon 129, there are 2 variations. One of these is part of a sequence called the M allele. The other variation makes a sequence, the V allele. Some researchers genotyped a number of different groups of Foray people at this particular locus, and they found these numbers of individuals with these different genotypes. Now, on the face of it, it's not obvious what the pattern in these numbers is. And that's because we don't have anything to compare them to. What Hardy-Weinberg equilibrium gives us is it gives us something to compare these to. It says if there's nothing wrong, we ought to see this. And if what we're actually seeing is different from what Hardy-Weinberg predicts, then we get a clue about what is actually happening genetically in this population. So how do we calculate what this population's ought to look like if nothing interesting is happening? What I'm going to do is I'm going to work through this group, Group A, the children who died from Kuru in the epidemic. Then I'm going to ask you to work through these next 3 on your own. You'll be telling me what results you get in the quizzes. What you want to do is go ahead and work through Group B and take the first quiz. If you get that right, then you know you're on the right track. Go ahead and practice C and E. The reason I want you to do this 3 times, which may seem very repetitive, is because this is something you want to get down to the point where it's reasonably automatic. Okay, this isn't something you want on an exam question to have to be really thinking very hard about. I want you to be able to get to the important step, which is where you take the expectations from these Hardy-Weinberg equilibrium calculations and you make predictions based on those, because that's why this is useful. That's when things get really interesting. So let's go ahead and take a look at the data from Group A. What do we do with these data? The first step when you're solving a complex problem is to know where you are going. With respect to Hardy-Weinberg equilibrium, there are 2 things we know we need to check. We need to ask, have allele frequencies change over time? And we need to ask, are the genotype frequencies in the ratio p squared to 2pq to q squared? Well, what we've got here aren't genotype frequencies, but they're numbers of individuals of different genotypes. It seems like what we really need to do here is get P and Q, the allele frequencies, for each of these groups. From there, it's really pretty easy to calculate what P squared, 2pq, and Q squared are. And then we want to be comparing those allele frequencies to what we see in these 4 groups, and these genotype frequencies to what we see within each of these 4 groups. So that's where we're headed. Now, do we have the information that we need here to figure out what P and Q are? Yes, we do. So let's go ahead and do that for Group A, and then you'll go ahead and do that for Groups B, C, and E. Well, we need to begin by figuring out what P and Q are. And we've talked about how to do that already using a probability tree. Hopefully by this time you've practiced this enough so that it's relatively automatic. If you take the number of MM individuals, they are going to donate M alleles with a probability of 1. VV individuals donate a V allele with a probability of 1. And MVs donate each one with a probability of 1/2. That means for the M allele, you take the number of MMs, that's 22, plus half the number of the MVs, that's going to be 6. Add them together and you get 28. Okay? Down here it's going to be similar. 14 plus 6 equals 20. Frequency of the M allele comes out to 0.58. The frequency of the V allele is 20 divided by 48. That's 0.42. Something I always do once I get to the end of these calculations is I take a look at these numbers. Do they make sense? Okay, well, we've got 22, that's bigger than 14, but not by all that much. Okay, 0.58 is bigger than 0.42, but not by all that much. That looks good. These 2 numbers have to add up to 1, and they do. Okay, it looks like we're ready to move on. Now we take a look at the genotype frequencies that we would expect to get if this population were at Hardy-Weinberg equilibrium. And this also should be pretty automatic. It's just picking 2 alleles out of the bag because one of the assumptions of Hardy-Weinberg equilibrium is that mating is random. So P squared is simply going to be 0.58 times 0.58. That's equal to 0.34. And it's really pretty easy to calculate these other 2 here. We expect 34% of our individuals to be MM homozygotes, 49% to be heterozygotes, and 18% to be VV homozygotes. Okay, do we actually see these percentages, these frequencies among the genotypes? Well, it's hard to tell, because down here we have frequencies and here we've got numbers of individuals. So what we probably want to do is take these frequencies and convert them into expected numbers of individuals by multiplying each one of these by 48. Okay, so when we go ahead and do that, we would expect to see, if these are the true genotype frequencies, we would expect to see 0.34 times 48 equals about 16 1/3 MMs. We would expect to see a little over 20 heterozygotes, and we expect to see a little over 8 VV homozygotes. Okay, let's compare these numbers to these. Well, it's pretty obvious what kind of pattern we've got here. These are relatively small numbers, so a difference of 6 in this case is a pretty large one. This number is higher than we expect. This number here, this number up here rather, is much lower than 20.16, and this one again is higher than what we expected here. So in this group, in group A, we've got an unusually large number of homozygotes and an unusually small number of heterozygotes. Okay, you should be able to follow these steps for groups B, C, and E yourself. Go ahead and take the quizzes that check your work. Be sure to check your work on one group before moving on to the others. And when you're done with all of those, you'll find yourself at another video which will explain how to make sense out of your results.